title of this poster is Rumen Metabolites of Periparturient Dairy Cows Varying in SARA Susceptibility Modify Bacterial Composition and Fermentation. Although postpartum dairy cows are fed with same diet, they may differ in susceptibility to developing subacute ruminal acidosis. One of the reasons for this inter-animal variability is thought to be due to the microbial composition in the rumen. The microbial composition of the rumen determines the type and amount of metabolites present in the rumen. However, vice versa, it is possible that the metabolites present in the rumen changes the microbial composition. In this in vitro study, we tested this hypothesis. Metabolites produced by microbes may alter the microbial composition and their metabolic capability. This hypothesis was investigated using sterile metabolites obtained from two groups of dairy cows during the transition time, SARA susceptible and unsusceptible groups. These two groups responded differently to the early lactation diet, which are rich in rapidly fermentable carbohydrates. The SARA susceptible cows experienced a decreased rumen pH and developed SARA during at least 2 days in the first 3 weeks after calving, while the second group of animals were unsusceptible to SARA development. Microbes obtained from 3 cows not suffering from SARA were exposed to the supernatant of each of the SARA susceptible and unsusceptible cows during in vitro incubation at normal, which is pH 6.8, and low pH condition, which is pH 5.8. After 24-hour incubation, changes in the bacterial composition as well as short-chain fatty acid production and lactate accumulation were assessed. When considered the results, the gene copy numbers of the bacteria were higher in pH 6.8 than pH 5.8, irrespective of the origin of the sterile supernatant. When considered the beta diversity, per MANOVA analysis revealed differences in beta diversity between inoculum before incubation and the bacterial community after incubation at both pH 6.8 and 5.8. But there were no differences were observed in bacterial beta diversity related to the origin of the sterile supernatant at both pH after incubation. However, we also observed changes in the relative abundance of some of the bacteria at phylum and genus level. Briefly, lower relative abundance of firmicutes, uncultured Ruminococcaceae, Lactospiraceae, and Mohibacterium, as well as a higher relative abundance of Prevotella, was found in sterile supernatant from SARA susceptible cows compared to SARA unsusceptible cow, irrespective of pH. When considered the net lactate accumulation of SARA unsusceptible cow at pH 6.8, which is higher than the SARA susceptible cows, but when considered and we didn't observe any differences between groups when microbiota were exposed to at pH 5.8. Compared to exposure to supernatant from SARA susceptible cows, microbiota exposed to supernatant from unsusceptible cows produced or tended to produce more volatile fatty acids. The acetate proportion was higher after exposure to supernatant from SARA susceptible cows compared to unsusceptible cow at pH 5.8, whereas the propionate proportion tended to be higher in pH 6.8 in SARA susceptible cows. As a conclusion, significant modification in some fermentation parameters such as VFA production, lactate accumulation, and volatile fatty acid profile was observed, And also changes were observed in bacterial composition at phylum and genus level based on the specific metabolites used in the in vitro incubation. Thank you.